Okay, as usual, let me do a recap of where we left off last week. We are last chapter. So far we've learned about how we inherit DNA, alleles and genes from our parents and through generations. The point of this topic is to investigate how the proportion of various characteristics can change over time. Yeah? Why is it the OG population look this way? After millions of generations, they now look another way across the population. We learned that there are two mechanisms that takes place. They can cause this change in proportion of characteristics over time. The first one we learned is natural selection. In natural selection, we start off with a population of organisms that inherently are different. There is variation in the population. Some of them have this characteristic, some have others. But it's because we have variation that there will be differences in the ability to survive. But the difference in ability to survive will only really show if we have something called a selection pressure. A selection pressure is some condition that will either jeopardize or increase an organism's chance of survival. Some traits may have what we call a selective advantage. It means that this characteristic has an advantage in this particular setting. If an organism has a selective advantage, it will be selected for. If it doesn't have the selective advantage, it will be selected against. Okay, so that's where we stop for natural selection. And then we also looked at a more artificial form of selection. In artificial selection, I ask you the same questions. Is there selection pressure? What is considered selectively advantageous? We realize that actually in the case of artificial selection, it is the human that's the pressure. We kind of decide who gets to mate and who gets to pass on genes to the next generation. And therefore, what is the selective advantage in the case of artificial selection? It is really subjective. What may be good to us may not be good to that poor dog with the flat face, the bulldogs. What may be good to us may be not so good to that poor dog that is really small. Actually, look at all the poor dogs, all the different breeds. Their body proportions, right, look cute to us. But I tell you, if human beings be selectively bred to cuteness, may be very odd also. Some humans, very long center part of the body. Right? Those dogs, they have very long body, right? Can you imagine if humans were selectively bred down different parts to accentuate different characteristics? Prefer people with big nose, or they selectively breed. Then you'll see people with very big noses. So when you look at dogs is a good example of artificial selection at work, but it's really subjective. What is good in humans' eyes may not be actually selectively advantageous if I put this dog back in the wild. So if I put a dog with a flat face in the wild, it's going to be out of breath the moment it starts running, because it doesn't have enough face to breathe. Yeah, as we saw last week, a lot of these bulldogs need to have corrective surgery so that they can even eat, so that they can even breathe. Remember that last week that poor bulldog needs to cut a hole here just so that he can breathe, because his face too flat. Yeah, who have got a flat face will be so bad. Okay, and that's where we left off last week. But one thing is in common between natural and artificial selection. We need many generations for us to achieve this. Natural selection will take many years. Repeated rounds of selection until the proportion of the characteristics change. Artificial selection also, even though it's controlled by humans, it takes lots and lots of years to achieve the particular apple breed we want, particular dog breed we want. The dog breeds we see today took millions millions of years to reach today. Okay? So both need many generations and time is often an issue. The whole point of today's lesson is to see how we can skip all that. What if we can skip the many generations and skip the time? Is there a faster way to change the characteristics of an individual? And that is where today we go into genetic engineering. The word genetic means they use the genes. Engineering means that we are going to now directly temper and readjust the genes to our liking. That's the whole point of today's lesson. And with that, may I get you to go to tab B? This will be the last time ever you use SLS. Unless you take bio and SH. Oh, yesterday, first time I hear that you never take bio in junior high, you can still take bio and SH. But yeah, it will be quite hard. I think the talk yesterday was very useful. Although I don't know how you all can hear anything. But the changes that are happening, I think you are very lucky. Yeah, it's just nice if you are that batch. The point of all the changes, if you see, really we're trying to give you more time off. Yeah? We often get caught up in preparing for exams, then we have less time and we rush, learning becomes very painful. Okay, the learning outcome is very simple. I feel like I'm missing a few things. Okay, we're going to learn how we can use genetic engineering as a technique to modify the genes of plants and animals. In LA, do you have a conversation about genetic engineering? I felt like you did. Was it part of your LA? It's job, is it? So you all didn't deal with the ethical implications of modifying genes. No, okay. Okay, so what do I expect you to know by the end of today? By the end of today, I hope you can take away the toolkit of a genetic engineer. What are the things you need to be able to genetically engineer an organism? After that, I'm very glad to take questions from all of you, because once you know how, then we should talk about Shubida. Okay? I think even before we reach this chapter, Kyra was already asking the question whether we should be modifying individuals. Okay, so I expect you to be able to, number one, list the tools required to transfer genes between cells. So for example, I have a bacteria cell and I have a human cell. You can, by the way, transfer human genes to bacteria and get the bacteria to produce human proteins. We actually do it on a large scale. For what reason? I'll share with you in a while. Number two, so I expect you to also know what the tools are. Actually, the tools, a lot of it are enzymes. So I expect you to be able to know what the enzymes are, how to use it. With that, let's scan through some of the questions first before we watch the video. First and foremost, first question I'd like you to answer as the video goes by. What are the molecular tools needed for gene cloning? When you hear the word gene cloning, literally means what it's saying. How can we make many copies of a gene? That's what gene cloning means. This is unlike replication. Replication occurs within a cell. But I'm asking you, how can we create many copies of the gene, like a photocopy machine? Okay? Next, which class of enzymes do restriction enzymes belong to? Okay, you will hear the word restriction enzymes. I'd like you to find out what kind of enzyme it is. You see the spread of enzymes here? They are enzymes we have learned before in our year 3. Okay, so question 1 and 2 are questions that we're going to work with. Ready?
First step in cloning a gene is to isolate the DNA. First step in cloning a
Before I start the video, perhaps I paint the context of why we want to clone genes. At the end of the day, when we want to genetically engineer an organism, we would want to have many copies of the gene so that we can, genetically modify many cells at one go. Yeah? Can you imagine a multicellular organism? If I only send one copy of the gene, I can only modify one cell. Make sense? So I need to clone the gene, have many copies, before I can really change individuals. Okay? So that's the intent of gene cloning. It's a photocopy machine so that I can give the instructions to every cell in the body so that they can all be changed. And what are the tools for that? Let's learn it now.
First step in cloning a gene is to isolate the DNA from the organism that contains the desired gene. The isolated DNA is purified and then fragmented with a restriction enzyme. Restriction enzymes used in cloning produce staggered cuts in specific sequences in the DNA, generating fragments with cohesive ends. Each fragment has a single-stranded sequence of nucleotides on its ends that is capable of hybridizing with DNA that has been fragmented with the same restriction enzyme. The DNA fragments are then incorporated into plasmids. The type of plasmid used for cloning has a single restriction site, and when cleaved by the restriction enzyme, generates the same cohesive ends that are in the fragments of the DNA to be cloned. The cohesive ends of the plasmid and DNA fragments now line up, and the enzyme, DNA ligase, is used to form phosphodiester bonds. The next step is to incorporate the plasmids into bacterial host cells by transformation. Each cell contains a different segment of DNA from the original organism. Taken together, these cells represent a DNA library. The cells can now be plated out on an agar medium. The colony of cells containing the desired cloned gene can then be identified and isolated.
Okay? Can we play the video? Questions 1 and 2, yeah?
First step in cloning a gene is to isolate the DNA from the organism that contains the desired gene. The isolated DNA is purified and then fragmented with a restriction enzyme. Restriction enzymes used in cloning produce staggered cuts in specific sequences in the DNA, generating fragments with cohesive ends. Each fragment has a single-stranded sequence of nucleotides on its ends that is capable of hybridizing with DNA that has been fragmented with the same restriction enzyme. The DNA fragments are then incorporated into plasmids. The type of plasmid used for cloning has a single restriction site, and when cleaved by the restriction enzyme, generates the same cohesive ends that are in the fragments of the DNA to be cloned. The cohesive ends of the plasmid and DNA fragments now line up, and the enzyme, DNA ligase, is used to form phosphodiester bonds. The next step is to incorporate the plasmids into bacterial host cells by transformation. Each cell contains a different segment of DNA from the original organism. Taken together, these cells represent a DNA library. The cells can now be plated out on an agar medium. The colony of cells containing the desired cloned gene can then be identified and isolated.
Okay, many things were brought up. First and foremost, we want to identify some of the tools that we saw or heard of within the video. Before I get you to click on the options that you heard in the video, what are some of the tools you heard of Second, what in the world are restriction enzymes? Okay, so let's have a look. What are some of the molecular tools needed to clone genes? Make many copies of them. Number one, we need cloning vectors. What is a cloning vector? The cloning vector is, in this case, this particular circular ring. Earlier on in the video, you have seen that bacteria contains a circular ring of DNA. So this is DNA. That is circular. We call such DNA plasmids. Or plasmid for singular, plasmids plural. Plasmids are what we call cloning vectors. A vector, usually, if you hear the word vector, it means that it will hold on to something. Okay? Or kind of like a hose. So these plasmids will hold on to the DNA. And we will see what I mean in a while. So here I also have a plasmid. This is a vector. Next thing, it says that we need restriction enzymes. Well, if you think of the plasmid as like a photocopying machine, we must somehow be able to put what we want, the gene, into the photocopying machine. Okay? We rarely ever photocopy the entire DNA of a cell. We tend to only want to photocopy what we want. Let's say this red section here is the DNA that I want to photocopy. So, logically speaking, therefore, to save the effort, I'm going to cut. Let me make a snip here and another snip over here. Okay? Minimally, we need to make 2 snips, correct? We call these enzymes restriction enzymes. Their role is to really cut out the gene, or what we call, sometimes you see GOI because it's referring to the word the gene of interest. This is what we are interested in. How might we insert this gene of interest after cutting it out into the plasmid? And you have seen in the video, you need to make one more cut. You make one more cut in the plasmid such that you can insert the DNA in. So this plus this, you end up getting okay. So how many cuts do you need to make in total? 3 cuts. 2 cuts to take the DNA out. 1 cut made in the plasmid so that I can insert the DNA of interest into the plasmid, my photocopying machine. Some students ask, what exactly is a plasmid? Is it made of protein? No, it is DNA. Okay? We are putting it into the DNA of bacteria so that when the bacterial cell starts to carry out its own version of mitosis, cell division, it will end up copying these plasmids repeatedly. So essentially we are hijacking the bacteria, making the bacteria do the cell division and help to create many copies of these plasmids. Because bacteria divide very fast, human cells divide very slowly, right? But because bacterial cells are within a few hours can have millions of them, wow, we just need to put one gene of interest in one plasmid into this bacterial cell. Within a few hours, you can have many copies. And after that, we will just need to kill the bacteria and extract out the photocopy. It's brutal, no choice. So in other words, we have scissors, we have the photocopy machine, one thing that is lacking, how in the world did I manage to insert this in? This is what this last enzyme is for. This last enzyme is like a glue. Okay? We need enzymes called DNA ligase to help to stitch together the bacterial DNA with the gene of interest, so that it becomes one continuous loop. And that's all the tools there is. We only need scissors, which are the restriction enzymes. We need cloning vectors, which are the plasmids, which we will insert the gene of interest to. And then we need a form of glue to stick everything back together. That's simple, only need three things. What I need you to know is what you already know from JH3. When these scissors are, so here's the clue up, when the restriction enzymes digest the DNA here, what bonds are you breaking? So that is the question I need you to answer. What bonds are broken? So in order to cut the DNA, okay, everyone have a think. What bonds are broken? Okay, then you must understand the structure of DNA. DNA is double-stranded, correct? Okay, so let's zoom in. I'm going to now zoom in. Let's say we want to cut this region over here. Okay, let's zoom in. This is the region I want to cut. Okay. So I ask you, if the scissors, the restriction enzyme were to come in, cut. Question is, what bonds am I breaking when I cut? This is a JH3 content. Yeah. Okay, MCQ lah. You either choose hydrogen bond or phosphodiester bond. Which one will you? Okay. The bonds between all the nucleotides, that's phosphodiester bond. Okay, so please take note. What are we cutting? We are cutting phosphodiester bonds. Okay, please take note. And therefore, if I want to glue things back up, what bonds am I forming? Forming the same bonds in order to glue them back. Okay? Last but not least, in your toolkit, I'd like to introduce you to two kinds of scissors. There are actually two kinds of restriction enzymes. The first kind of restriction enzyme, this is what they do. Very simple. They just cut literally like that. The second kind of enzyme, restriction enzyme, little bit more sophisticated. Rather than cut just this way, it cuts down this way and then down again, kind of like a zigzag. But the outcome of these two forms of cutting is different. Because if you were to cut this way, you end up with two ends that are what we call blunt. So after cutting, this is what you get. GC, then GA, CT, TCAG, we call such ends blunt ends. But it will make more sense why I call it blunt ends if we look at this context. If I cut my DNA this way, what will it look like at the end of the day? It will instead look a little odd because there will be an overhanging portion. So this is what it looks like. You will find that there will be a CTAG here, and then the other side of the DNA that will detach will go like that. Yeah? There is like an overhanging portion. We call such ends sticky ends. Or why call them sticky? Because remember in JH3, we learned that between nucleotides there's hydrogen bond. Remember? Well, if I produce sticky ends, it makes it more interesting. Imagine now, if I were to cut the plasmid here with the same restriction enzyme, I would then be able to, like a puzzle piece, insert it in, and it can stick to each other. Blunt ends aren't as good for specific kind of sticking.
First step in cloning a gene is to isolate the DNA.
Earlier on in the video, I'll show you again the sticky ends. So we look at the screen. Here, the enzyme, the restriction enzyme is about to make a cut that will end up with sticky ends. And it comes with an overhang. This overhang is really good. Because now, if I go and make the same cut in the plasmid will also have some overhanging parts. If the plasmid has overhanging parts, and the DNA we just cut out has overhanging parts, it makes it really like a puzzle piece that you can just stick together.
Each fragment has a single DNA that has been fragmented with the same restriction.
Here, you see the gene of interest. You find that if you were to cut both ends to produce sticky ends, you will have overhanging parts on both sides of the gene of interest. Then, we can place this gene of interest
The type of plasmid used for cloning
inside the plasmid. So watch out, when I cut the plasmid, I also produce sticky ends.
Cohesive ends that are in the fragments.
Now, I can put the gene of interest in.
The cohesive ends of the plasmid and DNA fragments now line up, and the enzyme DNA ligase is used to form phosphodiester bonds.
Okay? And that's it. Yeah. These are the toolkit for genetic engineering, how to transfer one gene and insert it into another organism's DNA. Which is better? Should we use restriction enzymes that produce sticky ends or those that produce blunt ends? Actually, there's one more benefit of having sticky ends. It makes it a little bit more specific. You look over here, technically, if I had used a different restriction enzyme that produces a different sticky end, this wouldn't be complementary to what I have here. So what does this mean? This enzyme here and this enzyme here, you better make sure that you're using the same restriction enzyme. Because if you use two different enzymes that produce two different versions of sticky ends, then it won't be complementary to each other. Okay? And that's usually the trick. We may give you a whole bunch of different restriction enzymes. Then we ask you to choose which ones you want to use to cut the DNA out and to insert the DNA in. Some students choose two restriction enzymes. You cut here with one kind, you cut here with another kind. But it's not going to match up at the end of the day if you use two different kinds of restriction enzymes.
The next step is to
Okay, so I take a pause here. Yes. So actually blunt ends at the end of the day, we could still insert it. Let's say this is the plasmid ends. We could still insert a gene of interest here with blunt ends. Okay, but the challenge with blunt ends is that there is no specificity. When you create specific sticky ends, then it can be very specific. We can be very sure that this is what we want to put in. But with blunt ends, we sort of don't have control. If a random DNA comes, it can also be inserted in. Yeah, so blunt end, you have less control, less specificity of how or what you want to put in. But if we make sure that we create the same sticky ends on both sides, then we can be very sure that whatever we wanted will be the one that is inserted. Okay? Okay, let's try question 3. They say a scientist wants to cut out a piece of DNA and to piece it into the cloning vector, which is the plasmid, depicted below. So we want to put it inside the plasmid. They ask you, both the DNA have restriction sites recognized by restriction enzymes. Restriction sites are the areas where the enzymes can cut. So which of the following processes will allow the scientist to create recombinant DNA? Recombinant DNA is the DNA that now has the gene of interest inside. It's called recombinant because we recombine recombinant DNA. So which is the correct answer? Can we give it a go? How should we cut to ensure that we can create recombinant DNA? Okay, everyone give a click. Let me see what our responses are. Okay, generally speaking, there are two correct answers. It's either A or D. Okay, many of us are choosing in that zone. Yes.
But you say it's not as specific, but technically still can.
Can still can. Yeah.
Like the options.
Oh. Okay. Okay. But when they say recognized by restriction enzymes A and B, I think they are assuming here that both enzymes produce sticky ends, just different kind of sticky end. Yeah. I think perhaps when you saw restriction enzyme B, you're thinking that it will produce one blunt, one produce sticky. Yeah. Yes. So say again, is there random DNA? Because as you are, yes, there could be random pieces of DNA, but also I think for efficiency's sake, if you have, let me think up. Why make it so specific? That's a good question. Why make it so specific unless, yes, there could be other DNAs floating around? Okay, so assuming we have more than one gene of interest, that we're interested in. Having this method will now allow me to piece together different restriction and different gene of interest into different plasmids. So for example, let's say I have 3 gene of interest. Here's number 1. Maybe I have another gene of interest here. And let's say I want to specifically put this one in plasmid 1. And I want to put this one in plasmid number 2. How can I ensure that this will join to this and this will join to this? Well, if I cut this out with restriction enzyme type 1 and this one type 1, then they will be complementary in stickiness. Then you definitely bind here. This one, I cut with a different restriction enzyme. Use the same one to cut here. Then it will be complementary in stickiness to this plasmid only. This gene of interest wouldn't be able to join here because it's not complementary in stickiness. Yeah. So I think it works, perhaps, it makes more sense in context if we have many gene of interest, many different plasmids that we want to insert in. Okay, so which of the following process will allow the scientist to create this? So A, cut both the DNA of interest and the cloning vector with restriction enzyme A. That's possible. If you cut both with the same restriction enzyme, they will be the same kind of stickiness, then they will be definitely be able to join with each other. So no problem with that. D, cut both the DNA of interest and cloning vector with both enzymes A and B. What is it saying? Community form, blah, blah. Is this possible? Okay, wait, I feel like this answer is wrong. How can you cut DNA of interest and cloning vector with both restriction enzymes? This is only possible if you do it separately. So let's say you cut the DNA of interest with A, then you cut the plasmid with A, then definitely you will join. You could also use it for B, but you must use B only in both instances. So I think this answer can only be correct if you make sense of it in terms of I can use both just A to cut both sides, or I can just exclusively use B to cut both sides. You cannot use both A and B at the same time, it makes no sense. Okay? If I use A to cut the DNA out, then I use B to cut the plasmid, it will not match like a puzzle. Okay? So this answer cannot be interpreted that way. You cannot use both at once, but you can use both exclusively. Okay, so that is the first part of today's lesson. We learned about the tools required for genetic engineering. Now we go into the case studies. What are some examples of organisms that we have applied this technology on? Because if you think about it, these tools that I just taught you, we only use it to insert the DNA into small little bacteria. But actually, if we wanted to, what's stopping us from inserting into other organisms? Yeah? So, same tools, you could use it for other purposes too. Here are some examples that we've already done. I'm not sure if you learned this in geography before. Things like, what is this called?
Wonder rice.
Yeah, you all learn wonder rice. The rice come out yellow color or something like that. Then there's also the special corn. Yeah, I think a chapter in geography deals with that. What are the history students here?
Yes, finish the exam.
Okay. What is he going to do the exam?
Exam that had the topic of
Okay.
Genes stand for genetically modified. So GM crops are farmers growing for food whose genes have been altered using genetic engineering. some cases, genes of other organisms have been inserted into the genome of the crop plant to improve it in some way. As the human population increases, there is a higher demand for food, and it's often difficult to grow enough food to provide everyone with a balanced diet. Growing GM crops is a partial solution to this problem, because GM crops tend to have higher yields than conventional crops. They can also offer extra nutritional value. Let's take a look at a few examples of genetically modified crops so we can see how this is possible. Crops that are most commonly modified are cotton, soybean, maize, and canola. Crop yields are often negatively affected because pests like insects damage the crops. Traditionally, farmers have sprayed their crops with pesticides to kill the insect pests, but this is time-consuming, expensive, and often harmful to the farm workers in contact with the poisons. Also, pesticides kill useful insects such as pollinators. It can harm other animals that eat the insects that have been poisoned. Spraying the pesticides also damages soil and water in the environment. Crops have been genetically modified with a gene from a bacteria called Bacillus thuringiensis. This gene causes the cells of the crop plant to produce an insecticide in their leaves that kills insects that eat it. The genetic modification reduces the need for chemical pesticides. The growth of weeds in fields reduces the yield of crops because the weeds compete with the crops for water, light, and minerals in the soil. Farmers can spray their fields with weed killers, but this may also damage the environment or even the crop plants themselves. So crops can also be genetically modified to be resistant to weed killers. This means that only the weeds will be killed by the weed killers. This doesn't reduce the amount of chemicals sprayed on the field, but it does mean that higher yields can be gained. Have you ever been told that eating carrots helps you see in the dark? This is because carrots are a good source of vitamin A, which is essential for healthy vision. A common problem in some countries is a deficiency in vitamin A because people have a diet mainly made up of cereals like rice with few fresh fruits and vegetables. To overcome this vitamin A deficiency, rice has been genetically modified to contain beta-carotene, which is used in the body to make vitamin A.
It's the same in
This gives the rice a yellow colour, so it's known as golden rice. Its taste is not altered, but it contains extra nutritional benefits. Other examples of GM crops include those that grow in difficult conditions, such as drought or cold temperatures, and crops that are resistant to diseases. All of these genetic modifications mean we can either increase yields from existing farmland, or we can farm low-quality land, both of which help us to feed the ever-growing world population. But, so far, we have only looked at the benefits of growing GM crops. Many people are worried about possible risks associated with genetic engineering. For example, little is known about the long-term effects of genetic engineering, as GM crops haven't yet been around for long. There are concerns that GM crops could breed with other plants, which would spread the modified genes into other populations, and this could have a negative impact on whole ecosystems. These risks are the reason why growing GM crops is banned in many countries. Many people think that more research needs to be done to make sure they're safe. So to recap, genetically modified crops can help in increasing the amount of food grown. Research is being done all the time to investigate possible risks to human health and the environment. Although many people have opinions about genetic engineering and GM crops, we need to ensure that our knowledge is informed by reliable scientific research conducted by many different scientists.
Yeah, I'm
If you
curious.
like the video, give it
Would
a thumbs up.
you eat genetically modified food?
Subscribe.
I don't know of any in the market. Most of the things we eat in the market, in the supermarkets, they are artificially selected for. Artificial selection not the same as genetic engineering. But let's say we were to introduce one that's genetically modified. Would you eat it? Okay, so I'd like to hear from you. Would you eat GMOs? What are your views? Can you elaborate a little bit? Yes, no. Why? What are you worried about? What are your comfort zones? Second, what questions do you have about GMOs? I'll be very happy to answer as many as possible. Personally, I don't dare to eat. For reasons I'll explain in a while. What if this time we do it on humans? Do you still love your partner if you know that he or she was genetically modified to look perfect? Would there be a difference if your partner was genetically modified to look perfect versus plastic surgery? If that culture deems beauty to look like that, then everyone will end up. Some of your views, how you say it's okay. How you think it's okay?
Like, you see like, oh, last time the fruit all natural, but then now like a lot of processing, a lot of like plastic, but we never let also what, so
Oh, you're like, I mean, how bad can it get? Nowadays we got microplastics, not like there are any bad effects. So maybe this one's not as bad as we think it will be. Ella? As long as it has more nutritional value. Well, what do you elaborate? What do you mean as long as you have more nutritional value? Yeah, like the golden rice. So that means opposite. Is there an opposite to this? Because you say as long as there's more. Okay, so in general, Ella's okay with it because it would seem that we modify because we want certain good things to be present. Siqi is like no because it's not natural. Siqi, but it's not natural but it's good. So how? Will you elaborate? Why you say not natural, therefore I'm going to draw the line there? Okay, it's a bit scary. What part of it is scary to you? Okay, you don't know what will happen. At the end of the day, it's very molecular. We change things here and there. What exactly will happen if we consume it? Not exactly sure. Henry is a resounding yes. Why yes, Henry? I mean, if they engineer it to be that way. Yeah. True. What if I engineer a chicken to be very, very muscular, lots and lots of meat, so that you all have more meat on the plate? Still okay? Yeah, still okay, right? But this one is slightly different from nutritional value, right? I'm making the chicken very buff. So that when you cook the chicken, right, you go to Hainanese chicken rice store, right, you see jacked, right? Chickens hanging on the shelf. Shermaine says only if I need it. What does that mean? Oh, okay. So unless you really need whatever that this GMO has to offer, you wouldn't necessarily take it. So for some yes, but for some no. How come for some will be yes, some no? Why? Look okay, then you eat it. Don't look okay, I won't eat it. So the one that look okay is not the one that looks like fake food, it's the one that not okay. Oh, okay. So you're looking at from the visual point of view. If it looks unnatural, it looks fake, you won't do it. Teacher say yes if it provides us more nutrients. Yeah? Kind of like some of your friends here. Jeanette, yes. She feels that it's because this one also it can help to solve a bigger issue at large, food shortage. If yes, but only if it's proven to not alter human genes. Okay. So if we done enough tests, I kind of in this zone. I'm like, okay, I'll do it if I'm very, very sure you can prove to me that it's okay. Kara says, if food crisis happen, we won't have a choice. So Kara is kind of in a, right now we won't, but if comes the need, we no choice. Shreya, no, because it will look weird. I'm not used to them. Okay? These are kind of related your comfort levels now. What we looks exactly the same. Like the rice same color, but more nutritional value.
But it won't look the same.
It could, really could. Yeah? Just like how our blood types are all different, right? But we are still look like human. Yeah? So it's possible. Would you, if it still looks the same?
Yeah, I guess.
Okay. It would seem that visually, it's very important to us what the food looks like. At least what we think it should look like. Teacher says yes, it tastes better because it's been genetically modified. We might also already be consuming it, just that we don't know. She also feels that this one can solve a bigger problem at large. Food shortage, which is a very real problem. Shafika, depends on whether the modifications will cause harm to me. At the end of the day, for Shafika, it's about your personal health also. Finya, yes, because it ensures food sustainability, also thinking very big scale. Zuni, as long as it's yummy. And no one died from it yet. As long as you're not the first to die. Kiran, feels like it's okay to consume once or twice, but not too many times. Why is there a limit for yourself? Oh, okay. Okay, that's an interesting perspective. I never thought of it that way. Maybe we just don't eat so often. Alpha, no, my body may not be used to them. A comfort level thing, unless you try it. Ziwei, yes. Confirm already. Without a doubt. Last but not least, Nabila, she will eat GMOs as long as it's safe to eat, as long as the checks have been done. Then consider this. One of my worries, which I think scientists are also trying to solve, is let's say this is the DNA of the rice. Okay, this is the DNA of the rice, and we have the DNA of another organism that has something special that we want to insert into the DNA of the rice. My worry, which I think scientists are still trying to figure out how to overcome, is that when we insert something new in, okay, by right, if I insert, it's now longer because there's more DNA. My worry is that if we insert something in, we have no idea what the downstream broader effects are. Because this gene may produce a chemical A, but we have no idea if chemical A interrupts with, let's say, this part produces chemical B, this one produces chemical C, and B and C normally interacts with each other. We have no idea if A could do something else, other than its intended function, which is to provide new nutrients. The body is extremely complex. We've learned before how many enzymes can come together. For example, if you look at one-time electron transport chain, there are so many proteins in that chain reaction for you to just produce CO2, the nutrients you need. Imagine if ATP, I don't know, my brain fart for a moment. Can you imagine if this one thing, while producing something good, ends up disrupting a chain reaction? That is the part that is scary. We have no idea also if the chain reaction is happening, but just requires a very long-term scale for you to see the effect appear. And so for myself, I need to be convinced that very long-term studies have been done. Short-term studies are not convincing to me. Long-term ones can prove to me that, actually the body is coping and there aren't any widespread effects on other genes that exist in our body. Our body is actually very intricately balanced. It has evolved over many generations to be balanced in this way. So to suddenly change, we are not sure what the impact may be. So for myself, long-term studies are the way to go. Kai says, as long as research shows that it's safe for person. So will it eventually hit the markets? I believe eventually it will. Yeah? Because there are really real problems out there that we can solve just with this technology. Food shortage, make the crop grow faster and bigger. Nutritional value. People may not get to eat as much, but they just eat this one-time potato, you can get all the nutrients you need. Isn't that magical? I wonder where we'll draw the line. Will we start using it on ourselves? Will you agree to that? Will you accept that? This is a very simple question. I think a bit obvious. So another example of a case study is a special corn called BT corn, also has been genetically modified. Which organism is the gene donor and the gene recipient? So two terms are just, you may hear floating around when you hear genetic engineering. The first one is gene donor. The gene donor is the one where we take the gene from. Gene recipient is the one that receives the gene. So quite straightforward. So if you give it a vote, yeah, the bacteria is the donor, or the corn is the recipient in this context. So, hey, think about it, the corn may look the same, but it contains the DNA from bacteria. Are you comfortable with that? It is not bacteria, but it has the DNA that contains bacteria. Second one, pole 2. Okay, for this to make any sense, you do have to refer to your notes. If you look at your notes, there are a few case studies. Do I need to go to your notes? For you. Okay. Now it's exam setting period. You need to be very careful. Don't open wrong document. Okay. Let me unplug first in case I mess up. I don't want to reset both paper. Now notes. Okay, your chapter is, this is chapter what? 11? 11, right? 9. See chapter 9. Okay, give a quick read. I'd like you to look at chapter 9. Chapter 9, you scroll all the way down, we provide you a few case studies. I do not need you to memorize the case studies, but I do need you, if I provide you a new context, can you identify who's the donor, who's the recipient? Simple, simple. Yeah? So we look at this particular context, the salmon. Give you a quick read, and you tell me which is the donor, which is the recipient. Okay? Identify. Okay, let's go back to the question. So which is the donor, which is the recipient? Okay, so we see a spread up. But most of us chose A. The Chinook salmon is the donor, while the Atlantic salmon is the recipient. So you look at the case study, you zoom all the way in, you will find that at step 1, we extracted a particular gene called the growth hormone gene from the Chinook salmon. Later on, if you look down the road, then we insert it into the salmon. Okay? And then that salmon ended up growing much bigger because of this growth hormone gene. So as a student, I expect you to be able to identify which is the donor, which is the recipient. That's all. Okay? And right before this, we learned about the tools required to cut, to glue. Okay? I'd like to say that actually, in reality, we are using GMOs on a very large scale for medicinal purposes. In our previous chapters, do you still recall how our body regulates blood glucose? Do you remember the enzyme insulin? Yeah? Insulin helps to regulate your blood glucose levels, yeah? Do you know that for diabetic patients, people with diabetes, they need to have regular insulin injections into them, yeah, to help to regulate the blood glucose levels. But what I'm not sure if you know is, where did this insulin come from? Want me to guess? Where did you get so much insulin from? Say what? Oh yeah, just spot the whole thing. Actually, okay, in the past, we got it from an animal. In the past, we used to get it from pigs. Yeah? Imagine that poor pig. Somebody's extracting out perpetually insulin from this poor pig. Then we clean the insulin, then we use it in humans. But there are a lot of issues with that. Sometimes our body will reject the insulin because this insulin is a pig insulin. Second, different cultures, religions will react quite differently to the thought of putting pig insulin into our bodies. So currently we are actually getting insulin from bacteria. Except that this is not bacteria insulin. It is actually bacteria that is producing human insulin. So yes, actually we have genetically modified organisms before. And we're actually using them very frequently. We have already found a way to genetically engineer bacteria. And these bacteria are now special. They produce human kind of protein, human insulin. From there, then we pump that into people with diabetes. Unfortunately, the bacteria have a very sad story, because if this is the bacteria and this is their plasmid, after inserting the human insulin in, we will work them, make them produce insulin for us. They produce lots and lots of insulin. Other than that, the bacteria will also divide, which is great, because now I will have many factories producing insulin. But the sad part of this story, but you probably won't feel so sad after all, is that at the end of the day I need to extract the insulin. Yeah? So how do I extract the insulin? I would have to kill all the bacteria. So what happens is that we have a very large tank. We incubate, let the bacteria grow. Or these genetically GMO bacteria grow. They grow and grow and grow. Once we have a large population, produce lots of insulin, and we kill all the bacteria and filter and get the insulin out. Okay? So I don't know if you feel bad about that. Personally, I'm okay with it. Okay, but then all of this insulin that's left, that is what goes into humans. Are you comfortable with that? Because on one hand, some of us was not comfortable with eating genetically modified foods. But are you comfortable with how GMO technology is used in this context? What made you change your mind? Okay, so have a thought. What made you change your mind? How come this one you're okay? Is it because you're not going to eat the bacteria? Okay, so we have a few more minutes before we close out the lesson at 10:30. There are some sample questions I would like you to try. And if you scan down the questions, you'll find that a lot of the questions are related to restriction enzymes, about using the correct enzyme to cut to achieve a recombinant plasmid. Okay, may I get everyone in the remaining time to try out these questions? Once you try out the questions, you have a better idea at how we use the tools. In a while, I'll go through question 1 because I think the finding out 1, 2, 3 is a bit challenging. Okay, we take a short pause. Tomorrow I will go through the rest of the questions. This week I won't have lessons with you at all, right? Alright. Because next week, Thursday is Teachers' Day, Friday is holidays. Yeah. Okay, so tomorrow will be the last lesson of my term. I should be able to, I mean we are nearly done. So by tomorrow we'll be done with the syllabus. Okay? Yeah, I'd just like to go through this part first with whatever time we have left. Tomorrow we will continue the questions and then we will go through the rest. Okay, so how do you figure out the sequence of 1, 2, and 3? Actually, what are we trying to test you here? Your concept of complementary base pairing. Okay, so you want to see if you know that concept. So we see over here, if we use an enzyme that produces this sticky end after cutting, technically speaking, whatever is over here should have been complementary to us here. Yeah? So what would the sequence be? If this was AATTC, what should I have had here? It should have been a T, right? If I want to match this together, T, then a A, and a A. Yeah? Ha. I ignore the C because the C is still complementary base pair to this G. Got it? Okay, so that's the first puzzle to solve. If we solve this, that means over here it's the same thing. This would have been a T, T, A, A. Now can we solve 2? Yeah, so now we work backwards. 2 would therefore have to be complementary to 1. Because that's what we learned, it needs to be complementary in stickiness. So it is A, T. Yeah? What are we left with? We're left with 3. Actually 3 is one of the easiest steps to do also. Just complementary the top. So it's T, T, A, A. Okay? And if you notice therefore, if we cut using the same kind of enzyme, we will produce ends that are the same kind of sticky, then they will be complement each other. And we can be very sure that this gene of interest will go to the correct last gene. Okay? Okay, last 5 minutes. Just continue trying the questions. We're going to go through all the questions tomorrow. Okay, we do have time tomorrow. We do have time tomorrow. So maybe I will end off instead by answering some questions you left behind for me. How Yang asks, is it possible to put all nutrients into one fruit, make it a superfood? It really depends whether that particular plant or fruit has all the right enzymes, the right pathways present. Because at the end of the day, every nutrient requires a chemical pathway to produce it. Yeah? So it's not as simple as putting a nutrient in. You need to put in almost the whole pathway to set this up. Shemay asks, do they affect human development? Since we are what we eat, does it mean if I eat GMO crops, I may also become, whether I develop differently? I don't know. Yeah? The outcomes we won't know until long term. But if the question, which I don't think is what you're alluding to, but if people ever ask you, if you eat GMO food, can that DNA end up going to your DNA? What do you think? They are likely to think, biologically speaking, will the process allow it? Because actually, when you eat GMOs, it goes down the digestive system. By right, your digestive system will have digested the DNA also. Okay? So, by right, and then after that, it goes into your bloodstream. From your bloodstream, it's not that easy to end up into all your DNA. Because at the end of the day, what do we need? We need the tools for it. We need enzymes to be able to cut, enzymes to be able to glue. But we don't have those, then actually it shouldn't be able to go inside you. Yeah. So technically you are safe. If people claim to say you eat a GMO, you also get the DNA. It's actually not so, it's not entirely possible. Can GMO genes or food be able, okay, no. It shouldn't be the case. But if I genetically modify CT, then your next generation will also have that DNA. How many generations does it take for all characteristics to show after crossbreeding? This one refers back to artificial selection. How many gens are? Many are. Yeah? Remember the cow example, the super buff cow? It took like 10 over years to be able to get it. How do researchers know the long-term effects? That's the thing. I don't know. Right? If they want to rush out a report, like all of us last minute workers, or we want to yield results, we want to show people that we are doing some good, would they rush this? I don't know. Kiran asks, what happens if GMOs organisms go wrong? Well, the reality is that when things go wrong, and which often does in cats, the whole organism will die. Yeah. So ethically speaking, are you okay with the fact that many organisms will have died before the current state that we have? Finally, I want you to think, it's not that easy to genetically modify an entire organism. Because to genetically modify an entire you, I need your billions of cells to all have new DNA. It's not very easy. So actually the easiest way to genetically modify you is when you were but a single zygote. After sperm and egg join together, at the zygote stage, if I modify that and let that undergo mitosis, then all of your cells, billions of it, will be the same. Alternatively, I could modify dad's sperms and mom's eggs before they combine. So people, we don't tend to want to modify entire organisms. It's very difficult once they are adult. Okay, tomorrow we will really close out the chapter and close out your biology. Beyond that, we are going to start your revision already. But it's just that next week we won't see each other, so our revision only starts before we do it. Then I mentally prep you. Tomorrow I'll also come in with each stack of papers. Yeah? So those are revision papers. Bio paper won't be until quite later. I think there's still a month more when we come back. So you take your time, every week do one. Don't need to show me your set of holidays. I just want to ask, we like you to keep one set and then we time practice in class. There are 3 sets. Because I am not sure if you all have enough stamina to feel full exam quiz, because we only done quizzes. Bioprac. Bioprac comes 4 people. Okay? Okay, now I'm going to keep one set so that you can punch out yourself, get a feel of it. All of you all work very hard. I don't want it to be a case where just lack of experience sitting down for so long that is what sets you back. Okay, that's all. Now it's your CC PMF. Okay, thank you class.